quickly we can get through this. So first of all, if you have a Chromebook open, we close it. Second of all, I mentioned this the other day, but there are somebody, at least 2 people who, as of Friday, hadn't signed up for the AP exam. So make sure you do that. We are going to have our exam this Friday for Unit 1. I'm not going to be here on Wednesday. You'll have a study period. You'll have a bunch of stuff to do. And if you have any questions, you can track me down tomorrow or Thursday, or you're welcome to reach out to me like through email, and I'll get back to you. I just have appointments. I will probably see you, almost certainly I'm going to see you on Wednesday, because I'm going to try to be here. But I have an appointment in the morning, and I have an appointment in the afternoon, and so depending on how long my morning appointment goes, I'm not sure exactly when I'll get here. And then there's some things that I'm going to go over, that like logistical things that I'm not going to talk about until after the exam. But if something happened to your grade over the weekend and you're not sure what it was, it's not something that stressed you out or didn't seem like it made any sense, you're welcome to look through the comments here on today's module. This is what we'll talk about after the exam. It will probably be on Friday after the exam, but I'm not going to, only if everybody finishes it. And the exam itself, I mentioned this the other day, it's going to be humane. And by which I mean there will be some multiple choice questions and there will be some short answer questions, but it's not going to look and feel like an AP exam. It's just going to feel like a regular test in a class, which means those of you who are used to just sort of not being tricked by tests and test questions should feel right at home. It's not going to be designed to trick you. Going forward, as we start doing test practice, then I'm going to start incorporating that AP trickery more and more into the exams that we do in class. Not because I like it, but I mean, I don't need to say this, because you have to be ready for it, okay? But we'll start humanely. And then if you haven't checked Canvas, since I got everything that I had graded as of Saturday afternoon. So if you haven't checked Canvas at some point in the next 24 hours, check Canvas and just make sure that everything looks the way that you think it should. If it doesn't, then just hold tight, because we'll get to what might have gone wrong and how to fix it. But I'm gonna target that for after the exam. I want to make sure you're ready for the exam. Okay, so I would say, are there any questions? But I'm not going to answer any questions about that now. Instead, let's talk about the stuff that I asked you to do for homework. If you haven't turned this in, you can do that right now. You can actually, while I'm talking, you can open up your Chromebook, take a picture of your lipids questions, and then put them in the bin. You can take a screenshot of your 1.5 topic questions. The lipids, this isn't going to affect your grade either way. You can do it or not. But for all 3 of these, does anyone have any questions? Was there anything that came up that you were like, I don't get it, I don't understand what's going on? Yeah.
Were we supposed to handwrite them?
It doesn't matter. So the question was, were we supposed to handwrite them? So here's the thing. What I want is for you to learn this stuff, and if it's easiest for you to type stuff up, or like if you were, when you're doing homework, some of you use a digital tablet, that's great. You don't have to handwrite stuff if you don't want to. But there are going to be some things where, diagrams are important. So I'm not going to take points off if you did everything on a computer. But it might be easier for you to handwrite some of the stuff. To the degree that you, if you've done everything on a computer, then you're not going to have anything to put in the bin. So just do me a favor and put a comment in Canvas that says, I didn't do this on paper, so that when I'm going through and grading it, I know it's safe to look at your Canvas. Because otherwise what happens is I click on whatever you turn in, it's just a blur, I can't read it. So, but if you typed it, you don't have to print it out and put it in the bin. Sorry?
Do you have a handbag next week?
No, I just have everything here. And I will give it back to you. Yeah. Let's see, so Jax, did that address your question? Okay. All right, other questions about either the logistics of these three things or the content? Okay, the one thing I want to say is, I can't remember if this shows up. This is going to show up somewhere. I can't remember where. I think it might be in the Unit 1 study guide that most of you have probably already started working on. I mentioned before that some people claim that lipids are a polymer, and the monomer are those acids, the fatty acids. That isn't accurate. A fatty acid is not a monomer. They're not being hooked together to make longer and longer chains of the same thing. It is not a polymer. However, if you're asked, what are the monomers? The answer is fatty acids. You're not going to get asked that on the actual AP exam. You're not going to get asked that on my test because it's not accurate, but you'll see that online because people are confused. They don't understand the chemistry behind monomers and polymers. They just think of like if you take two things and hook them together, they must be monomers, and whatever you get must be a polymer. That's not accurate. Okay, and I'm just going to move on unless there's questions. Okay, if your Chromebook is open, will you close it? And you should grab something to write on and something to write with. This is the last chunk of information that you're going to need to know for the exam. And when we're done, I'll tell you a little bit more about the exam, what it's going to look like, and what's going to be on it. So we've looked at all of the major biochemical macromolecules now, except for nucleic acids. So that's going to be our goal for today, is to take a look at those. Most of you are going to find that you know about 80% of this, somewhere around 80% of this. So hopefully I'll be able to go reasonably quickly. So nucleic acids, the polymer for nucleic acids is nucleic acids. So remember how for proteins the polymer was polypeptides, and for carbohydrates the polymer was polysaccharides. They had kind of a different name for the generic polymer. Well, with nucleic acids, that is the name for the polymer. The monomer is called a nucleotide, and you're all familiar with nucleotides, even if you don't remember that you're familiar with nucleotides. You'll when you see them, you'll recall what they look like. There are a few different things that are made up of nucleic acids. One is DNA, or the extended version of that. That's just the abbreviation, is deoxyribonucleic acid, which I don't believe you're ever going to need to write. And you don't need to write it now. You just need to be able to recognize it if it shows up in an exam. I don't think that they would put it, but they might. And then RNA, which is ribonucleic acid. It's very similar, only it's just got a slight difference in one of the pieces of a nucleotide that leads to a slight difference in another piece of the nucleotide, and it's functionally used differently inside cells. And then the other thing that we'll see is some things that aren't used for genetic information at all, like ATP, and there's GTP and GNP and AMP, and these are all molecules that are used for either energy or signaling or both inside of cells. And you don't really have to worry about those right now, except to know that occasionally you might see something and recognize, oh hey, that looks like a nucleotide. And you're right. And that puts it into this category of molecules. But it's really, when we think about nucleic acids, what we're really generally talking about is DNA and RNA, because functionally those have to do with genetics and protein synthesis. All right. So let's take a quick look at nucleotides, the structure of a nucleotide. So there's three parts. There is a sugar. So oddly, this particular group, I mean, this is true for lipids to a certain extent as well, but this particular group, if we look at what it actually is, it's a sugar with some stuff attached to it. And the sugar is a 5-carbon sugar that's kind of the anchor of each nucleotide. And this sugar is either ribose, which is a 5-carbon sugar, or deoxyribose, which is a 5-carbon sugar. That's really close to ribose, and you don't need to know the differences, except you could probably guess, since it's ribose and deoxyribose, the deoxyribose is missing one oxygen atom. That's the like what you would notice. That's the noticeable difference between these things. And then there's a phosphate group, and you can draw this. This doesn't have an orientation in space, right? Like if we took a nucleotide and blew it up to a giant size, it's not like we'd look at it and be like, oh, that's the left-hand side. But conventionally, it's really typical to draw them like this. The sugar is in the middle, the phosphate group is off to the left, and then the last piece, which is a nitrogenous base, is off to the right. That doesn't have to be how it looks, but it's really common to be drawn this way. And these sugars are, if you remember when we were looking at carbohydrates, the sugars, the carbons in the sugars are numbered. And you can't really see them in these pictures, but the way those sugars are arranged here is there's one here, carbons, there's one here, there's one here, there's one here, there's one here, and there's one here. And so, so when you look at these pictures, these little pentagons, the tendency is to think that each one of these points is a carbon, but it's not. That's an oxygen there. Carbon, 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 and the 5th carbon is out here. Just so you kind of have an image of what that sugar looks like. All right, when you start taking, yeah?
Did you say the oxygen was on the top of the
Mm-hmm.
middle one? And is it a double bonded oxygen?
Mm-hmm.
Okay.
Yeah. If you start taking these nucleotides and putting them together, you get nucleic acids. The one that you're most familiar with is DNA, and again, you don't really have to write that down. This piece you must already know. If you don't, it's like, don't panic, but this is knowledge that everyone, me and the people at the AP College Board are assuming you know already, that it's arranged in this double helix, which is basically like a ladder shape that's twisted. And we'll play around with that a little bit, not now, but when we start talking about genetics, we'll play around with DNA a little bit and work with models so that you can see physically how that all plays out. It is anti-parallel, and we'll look at that in just a second, and you'll get some ideas of what that looks like structurally. But basically what that means is one of the strands goes like when it's oriented vertically like this, which is typically how we draw it. One of the strands goes up and one of the strands goes down. But here's the thing, it is like there is no real spatial orientation. It's not like in a cell the DNA arranges itself so that those nucleotides have a left and a right, and DNA strands are always up and down with respect to the Earth's gravitational field. Like that's not what it looks like. So it's all by convention. By convention, we typically draw nucleotides with the phosphate group on the left and the nitrogenous base on the right. And by convention, we say that a strand of DNA has a direction. And we'll talk about that direction in just a second. All right, so that's good for now. So when we talk about anti-parallel, what that means is this arbitrary direction that we say is the direction that DNA molecules go goes one way for one of the strands in DNA and the other way in another strand. And this will have to do with the overall structure of the DNA molecule. So let's look at that just real quick. So first of all, DNA generally consists of two attached strands arranged in that double helix. it's possible to have single-stranded DNA, but in general, when it's in cells, it exists as a double helix, two strands joined together that are running anti-parallel. The two strands are attached to each other by hydrogen bonds between the nitrogenous bases. And we'll look at that in a little bit more detail later, but this is where you get the whole A bonds to T and C bonds to G thing. That's where the base pairs come in. That connects two molecules. These hydrogen bonds are pretty weak. The strong bonds are between the nucleotides of one strand and between the nucleotides of the other strand. So that happens through a covalent bond that joins the sugar group of one DNA and the phosphate group, of one nucleic acid and the phosphate group of another nucleic acid. So they're connected by the carbons on the sugar. And you can see that here. This phosphate group is kind of in between it, but really what's going on is this carbon on this sugar is connecting to this carbon on this sugar. They're connecting through the phosphorus group, through that phosphate group. And those bonds have a name. They're called phosphodiester bonds. And there's a reason for that, and you don't need to know it. Just similar to the fact that we call them peptide bonds and glycidic bonds in carbohydrates. All those different kinds of bonds have their own names. Okay? And then here's where the direction comes in. We say that DNA runs 5' to 3'. That's the direction of DNA, and you'll see in just a second specifically what that means. But the reason that we've chosen this is because it turns out that this is the way that DNA is constructed. It starts with 1 and then it attaches something to the 3' of that via its 5', and it attaches something to the 3' of this via its 5'. And this is the way that DNA is read when genes are used to make a protein. So we say that it has a directionality, and it's 5' to 3'. The 5' refers to the 5th carbon. The 3' refers to the 3rd carbon. So we'll see a close-up of that in just a second. And the anti-parallel part of it is because if you look at that 5' to 3', what you'll see is one of those strands has the way that I've drawn it up, or the way that it's depicted up here, one of those strands has its 5' carbon on the right and its 3' carbon on the left, and the other has its 3' carbon on the right and its 5' carbon on the left. And so because when we orient the strands so that they're running horizontally like this, one of them is going from 5' on the left to 3' on the right. The other is going from 5' on the right to 3' on the left. We say that they're running in opposite directions, or that they're anti-parallel. So here's what it looks like close up. Here's our sugar. So the carbons are numbered 1, 2, 3, 4, and 5. And if you look at where our phosphate group is attached and where the nitrogenous base is attached, you can see the nitrogenous base is attached at the 1', the first carbon. The phosphate group is attached at the 5', the 5th carbon. And the bonds between one nucleotide and the other happen here on one of them, between the 3' carbon of one nucleotide and the phosphate group, or the 5' carbon of another nucleotide. So you get this bond. The phosphate group is in the middle, but it's got this is the 5' carbon here, this is the 3' carbon here. 5' carbon here, 3' here. So you can see that it's running from the 3' carbon to the 5' carbon on this strand, going from bottom to top. This strand is running the opposite direction, and in this image, they've even put the letters upside down so you can see. This one is upside down. It's going from, this is the bottom, this is the top, so to speak. And it takes a little bit of time to get used to it. It's arbitrary. So, it's not like a book where you look at it, it's like, well, the words are clearly going from left to right here. It's like if you just found a bunch of shapes, and then you were like, I'm gonna say that this is the left side and this is the right side, and then boom, that's what you, from then on, that's what you call it. It's a convention, it's not, and there's a reason behind it, but it's not like if you take a DNA molecule and you throw it up in the air, it's gonna land with the phosphate group over here and the nitrogenous base over here. It's just not how it works. Yeah.
Do they normally not show the oxygen in like the diagram?
Yeah, they just don't. And that's, so it depends on what you're studying and why you're studying it. But for our purposes, usually it won't even go into this level of detail. You don't usually see the phosphate groups and the hydroxyl groups on here. Usually you just see like shapes and blobs and stuff. What we're most interested in is what's going on here. This is where we're going to focus because that's where the code is. Yeah.
How do you know if it's like the 5 and the 3?
So the way to do that is, I'll show you another picture in just a minute. That'll make it a little bit easier. But the way to do it is to take the nucleotide and orient it in space, either by drawing it or just visualizing it, so that the nitrogenous base is on the right and the phosphate group is on the left. And then you start numbering with the carbon where the nitrogenous base is attached and go counterclockwise and then off. So you have to know what the molecule looks like when it's organized in the conventional way. And then you know which carbons are involved with the connections. Okay, let's talk about the nitrogenous bases. This is also something that I think all of you are going to remember. There's 4 bases. I don't think you're going to need to know the actual names. You should recognize them. This shouldn't be a surprise to you, but you won't have to probably write them or identify that A is adenine, but it is. So our A base is adenine, our C base is cytosine, our G base is guanine, our T base is thymine, but that only shows up in DNA. And it's kind of weird because it's like, well, why does that only show up in DNA? And the answer is because the sugar in DNA is deoxyribose. And because it's a little bit different chemically, you can't attach the other base to it. And it's the opposite for RNA. RNA only has uracil. Oops, my bad. I should say RNA only. So RNA is only for uracil, and uracil is only for RNA, and it's just because of the different sugar. It won't attach. And it's just one tiny little atomic difference, but that's what does it. And so hopefully this is familiar to you. When we look at those base pairing rules, it turns out that there is a pattern to it that is has to do with the size of these bases and the number of hydrogen bonds that occur between them during bonding of one strand to another. So here's what it looks like. Adenine and guanine are a double ring. Thymine and cytosine are a single ring. Uracil is also a single ring. That's not pictured here. Thymine and uracil are essentially the same, really, really close to the same. And those styles of rings have names. The ones with 2 rings are called purines. The ones with 1 ring are called pyrimidines. But I don't think you're going to need to know that either. But you might. So I would commit it to memory. And the way that I've always remembered it, as stupid as it is 2 rings is bigger, but it has the smaller name. 1 ring is smaller, but it has the bigger name. That's how I've remembered it, literally since I was your age. And then of course, you have to know the words, but if you can remember purine and pyrimidine, then you can remember the one ringers are pyrimidines and the two ringers are purines. And you'll only get hydrogen bonds between a purine and a pyrimidine. And the way that these things are structured, they'll only bond with one of the other kind because adenine and thymine or uracil form a different number of hydrogen bonds than guanine and cytosine. So this is where the base pair rules come into play. Adenine will bind with either uracil or thymine with 2 hydrogen bonds, and cytosine and guanine bond together with 3 hydrogen bonds. And this is a cytosine guanine bond right here. You can see that they're hooked together with 3 hydrogen bonds. Okay.
So for DNA, for example, we have those 2 like parts that are connected for the ladder runs.
Yeah.
Are like one of those a purine and the other one's a pyrimidine?
Correct. Yeah.
And we can't have it where it's like 2 are purines and 2 are pyrimidines.
Correct. Yeah. And if you look at what's going on here physically, you'll realize it's actually the shape of them. You have one small and one big. It has to be one small and one big, so they take up the same amount of space. If you have 2 smalls, then the DNA molecule would shrink in that spot. The width of the ladder would get smaller. And if you had 2 purines, the double ring, the width of that ladder would get bigger. So it just turns out that it's a much more stable structure when it's built with 1 purine and 1 pyrimidine. All right. This is the last thing that we're going to talk about, and so this has to do with the differences between RNA, DNA, and RNA, and most of this stuff again should be reviewed. One has to do with the type of sugar in the nucleotide. Here's deoxyribose. Here's ribose. See that little oxygen there on the second? It's a hydroxyl group on the second carbon. It's a hydrogen in deoxyribose, and it's a hydroxyl group in ribose. That's it. And then because of that change in the sugar, that last base doesn't work with both RNA and DNA. So DNA will always have thymine as its 4th nitrogenous base. RNA will always have uracil. And if you look at thymine and uracil, you'll see those are really different too. There's a methyl group on thymine, and there isn't on uracil, and that's it. I mean, those look pretty similar at a glance. And you don't have to know what the structures of those are. I wouldn't know them if they fell on me. All right, let's see. So another difference is that DNA is typically double-stranded in that double helix. RNA typically exists as single strands. And again, that's something you probably already knew. And then they have different functions. And this shouldn't be a surprise. DNA stores genetic information. And that stays in eukaryotic cells, the more complicated kinds of cells, that stays in the nucleus, where it's a little bit safer, a little bit less prone to mutations. And RNA is used as a communication molecule. So, and we'll obviously talk about this down the road, but during protein synthesis, we take a section of DNA and make an RNA copy from that. That's called transcription. And then that RNA copy is read by a different kind of RNA and used to build a protein, and that's called translation. So RNA basically is like a messenger. It takes the information from the DNA and brings it out to the rest of the cellular machinery where proteins can be made. When we talk about genetics, we'll come back to this and we'll look at the structure of DNA in more detail, and we'll look at base pairs and codons and all that stuff that you almost certainly remember from biology in more detail. But for now, this is what you need to know. So Does anybody have any questions about that? Okay, so let's talk a little bit about the what's going to happen over the next couple of days, including the test. So first of all, there's a bunch of stuff on the module for you to work and think about, both for today and for Wednesday. So for today, there is an assignment on Canvas. It's the same style where you're just like writing the letters, nucleic acid, structure, and function. I'm guessing those aren't taking you too terribly long, is that right? Okay. And then there's some topic questions in AP Classroom. That's also not probably taking you too terribly long. There are just 3 of them per topic so far. The Unit 1 study guide I posted a while ago. Somebody asked if there's a key. There is. I posted it over the weekend in the assignment. So when you click on the assignment now, it will bring up the key. So you can check your answers, and you can also ask me. Send me an email, or just if like we're at school, you can track me down and ask me about it. The lactase lab, I said that I want you to do the introduction and methods. That is not due until next Monday, and it's not even due next Monday. Like, you're not going to have to turn anything in. It's just ideally you will have gotten that done. So most of you, quite logically, are going to put this off until after the exam, and that's fine. I just want you to know that it's out there. On Wednesday, here's what you're going to see on the module, and I haven't published this yet because you'll see this is like a working version of it. I'm still fleshing it out. First of all, there's going to be a quiz, not this week but next week, over column charts with error bars, and we'll have time to practice that with the lactase lab next week. That isn't going to be on the exam. You don't have to know about the error bars and the SEM. It may show up. You may see it while you're studying. You will see it while you're studying, but never fear, that's not going to be on the exam. The stuff that's going to be on the exam is going to be the stuff that is on AP Classroom, the content, the stuff about macromolecules, okay? And so on Wednesday, you'll have time to finish the study guide if you haven't done so already. This afternoon, I'm going to open up what they call the progress check. It's like a practice test on AP Classroom. This will be a assignment. If you finish it, great. If you don't, that's fine. I'm going to ask you to post a screenshot showing me, your scored version of it. If you don't get it done, though, that's fine. You can get it done or not. It'll be open starting today, and you can use as much of your time on Wednesday to work on that as you want to. There's a practice exam. This was written by Mrs. He. This is where you'll see some bar chart stuff. You can just ignore it, or you can practice it. It doesn't matter. It's not going to be on the exam on Friday. You do need to know it. So all of this stuff is here for you to work on. All of this stuff is, so the practice exam, I'm not going to have you turn in. The progress check and the study guide, I am going to have you turn in, but it's really up to you how much you do. And I'm not going to look through it and be like, oh, you didn't answer question 7 on the progress check, so you lose a point. It's up to you. What I want is for you to prepare for the exam. To the extent that these things are helpful for you, do them. If they're not, it's not a hoop that you need to jump through for me or for points. I just want you to learn this stuff and have lots of opportunities to practice. And then at any time during the period on Wednesday, you can just study. So you can work on these things, you can work on these things in groups, you can work on these things sometime, you can work on your own for other times. But what I want is for you to not be overwhelmed by the amount of stuff that I'm giving you, because I'm giving you, what I want to do is just make sure that nobody runs out of stuff if they just want to study. Like, if that's how you operate, I'm good with that. But not everyone necessarily is going to need to do it, okay? All right, does anybody have any questions? Yeah.
Do you have the printout
I don't, no. Yeah, just do it, so I know it seems like a chore. You're welcome to print it out if you want to. My opinion is that you'll get more out of it if you don't. And I know that sounds crazy, but that's been my personal experience, and it's been my observation as well. Filling in the stuff is less useful than looking at the sheet and writing it by hand on a piece of paper. You don't have to copy all the questions. Like, do what's useful for you, right? Like, it's fine with me if you just have an answer written down. It's what's useful for you. But what I have found is that it's to the extent that my work stands alone, I learn better. Which means, like, I often will draw the structures so that I can see them myself and draw them myself and really think about them and then answer the questions and stuff like that. It's not required, but it's a better way to learn it. Other questions? Okay, so we have officially 2 things due on Wednesday. The nucleic acid structure and function, and the 1.6 topic questions. We got 5 minutes. If you have questions, ask away. If you want to work on this stuff, knock yourself out. If you want to just do some logistical stuff, like get stuff turned in and put in the bin, that's great as well. And if your brain is full, then you can just sort of be done. Yeah.